Swayam Prabha. Digital India. Educated India.
Welcome back to my course Industrial Biotechnology. Now today I am going to talk about this fodder yeast production, and fodder yeast means the yeast that is used as a animal feed. And in the last class I talk about this baker's yeast fermentation process. Baker's yeast is usually considered as a food yeast, and food yeast is usually we also term as the nutritional yeast, because it contains about the 50% of protein, and also it contains vitamin B and other essential minerals, which is required for our as a nutrient. And I told you that two type of baker's yeast that is marketed. One is active dry yeast and this compressed yeast. And also I talk about this what are the different parameters that influence the baker's yeast fermentation process, and what should be the characteristics of the baker's yeast. One important characteristic I even mentioned that it should be well dispersed in the water, because if it is not well dispersed, there causes some problem in the bread making industries. And also I told that this baker's yeast also can be, one thing I forgot to mention, this can be also used for the production of yeast extract. With yeast extract, when you treat with chloroform under anaerobic condition, we keep it a high temperature, maybe 40 to 50 degrees centigrade, then soluble, the yeast cell will burst, and soluble material present inside the yeast cell that will comes out, and that we can use as a yeast extract. Now today I want to discuss the fodder yeast production, and fodder yeast basically this is produced by using another kind of yeast, what we call Torula utilis. Now the yeast is produced cheaply to feed animal is called fodder yeast. This has the good nutritional value. And when I was talking about the single cell protein, I told you that a more advantage of the single cell protein is that your cell must grow very fast as compared to any other biomass that is a plant biomass. So naturally that that it is always desirable that to use the microbial protein as our food. But this is not used in practice. The reason is that most of the microbial protein contains lot of nucleic acid, and this nucleic acid causes some kind of kidney stone and gout. So, so if you want to use this as a human food, then we shall have to we have to take the nucleic acid out from this single cell protein, then it can be used as human food. So here the Candida tuberculosis is mostly used. It has a good amount of nucleic acid, so it is not used actually for human food. It is used for animal food, and this is usually produced from the waste product from the industry, because particularly sulfite waste liquor from the pulp and paper industry. Now if you see that it is the microbial protein synthesized by yeast, in which the digestibility and amino acid contents is higher than the animal protein, and thus has a high biological value as compared to other feed. So this is one of the very important aspects why it considered as a animal food. Now the manufacture of fodder yeast is of interest to those desired to convert waste or surplus or low cost carbohydrate material into products of value for feeding cattle, hogs and other farm animals. The utilization of waste sulfite liquor from pulp and paper industry for fodder yeast production, as for example, results in the extension of animal food, at the same time aids in the elimination of stream of pollutants, because this sulfate waste liquor, if you throw it to the atmosphere, this causes the environmental pollution problem. This is another issue that we have that is to be seriously considered. Now, benefits of using yeast as a fodder is the inactive dry fodder increases the bioavailability of protein as they contain essential amino acids. The composition of this yeast in the following amount includes the 10 essential amino acids. We know very well that what is the purpose of essential amino acids. Essential amino acids are those amino acids which are not produce our body as desired amount. So, that is why we shall have to supply those amino acids for our for our growth and metabolism. So, that is very much important. On the content that amino acid, the fodder yeast is similar to protein of animal source. In the animal origin, we have all the amino acids more or less balanced, and feed yeast contains the vitamin B. In this respect, superior to all proteinaceous feed, including the fish milk. The B vitamin present in the yeast regulates the fat metabolism. This we have. The fodder is rich in vitamin D2. The 1 kg irradiated fodder yeast can bring the contents of vitamin D of 500 to 5,000 to 12,000 international unit. As of fodder yeast also contains valuable animals and birds micro and macro elements such as phosphorus, potassium, calcium, iron, magnesium, sodium, sulfur, copper, cobalt and others. Sulfur and its compounds are included in the composition, and biological processes involved in the formation of amino acids. The phosphorus and calcium, the composition is the yeast promote this normal development of skeleton. Overdose of fodder yeast does not cause any kind of side effect. Now yeast commonly used commercially is the strain of Candida utilis I told you just now, which also known as Torula utilis. The Torula utilis and Candida utilis is basically same, and often called is Torulopsis utilis. The other yeast used commercially or studied extensively also in the lab, this is Candida tropicalis and Saccharomyces cerevisiae. Now, advantages of using Candida utilis is that Candida utilis can utilize the hexose and pentose sugar, because if you look at this Baker's yeast fermentation process, your Baker's yeast Saccharomyces cerevisiae can only use the hexose sugar. They cannot use the pentose sugar. Now, when we use any kind of that plant material, it contains also not only contains cellulosic material but also contains hemicellulose. Cellulose is basically a polymer of hexoses, but if you look at the hemicellulose, it is a polymer of both pentose and hexoses. So, naturally that and in case of pulp and paper industry, we do some kind of acid treatment or alkali treatment, and during this process some of the hemicellulose that solubilize. So, there is every possibility of presence of pentose and hexose sugar. So, if you that is why we find that waste sulfite liquor, that is a good raw materials for the fodder yeast production, particularly for the utilization of Candida utilis. Now, the Candida utilis can utilize hexose and pentoses and other constituents found in the acid hydrolysis from the wood and other cellulosic material. As we know that pulp and paper industry, they use the wood type of material, particularly bamboos, for the production of paper, that it is particularly desirable on account of its high protein content and its vitamin B complex. Saccharomyces cerevisiae can use the hexose, I told you, disaccharides, but not the pentoses. But so it also can, Saccharomyces cerevisiae, it has the invertase enzyme. Due to the presence of invertase enzyme, sucrose is degraded to glucose and fructose. Candida utilis has got the meaty flavor. This is when dried. This is one very important aspect that we have when you dry the Candida utilis. Is give the meaty flavor, kind of flavor that is developed when you dried. Now, raw materials for production, it is desirable to use the raw materials with readily available but low in cost, because since we are using this as a fodder yeast, the raw materials should be low in cost, otherwise cost of production will be very high. On a commercial basis, sulphide liquor, molasses, wood sugar are used. However, hydrolyzed grain, citrus fruit or fruit juice, hydrolyzed olive residue and other materials when properly prepared also acts as raw materials. So, these are the different materials can be considered for the production of fodder yeast. Other resources like hydrocarbons oil, refined liquid paraffins, low alcohols like ethanol and methanol, hydrolyzed of wood waste like sawdust and chips, and distilled dry grain solids, distillers dry grains solid or solubles, the hydrolysis, then sulfite alcohol also be used. Now, ingredients for 5-inositol inosine monophosphatase or the guanosine monophosphatase, the IMPN. In Japan, yeast is the first extracted in order to remove ribonucleic acid content and leaving all protein intact. This ribonucleic acid is the source of two flavoring agents, namely that gualytic monophosphatase and inosinic the monophosphate. The remaining protein rich residue is used as high protein feed. This makes the fodder yeast much more cost effective. Now, raw materials for the fodder yeast production, I told you this is the waste sulphide liquor is largely used, which is the byproduct of the paper, pulp and paper industry. It contains considerable aroma of pentose sugar, and waste sulphide liquor contains 50% cellulose, 25% lignin and 25% of hemicellulose. Lignin and hemicellulose can be obtained by in dissolved state. The hemicellulose contains about 80% of hexoses like glucose, galactose, mannose, fructose, and 20% pentose like xylose and arabinose. Now, raw materials were in sulphide liquor, which obtained from pulp paper industry. This contained 8% dissolved solid, 1.5% sugar. I told you that 80% hexose and 20% fructose. pH is 2.6. That temperature is 76 degrees centigrade. It contains considerable amount of sulfur dioxide and toxic material. Also, it contains the suspended particles. So, as you know that sulfur dioxide is a strong oxidizing agent, and since it contains the sulfur dioxide, your yeast, when you inoculate in the media, they will not grow properly. So, if you want to grow the yeast cells, you have to remove that sulfur dioxide from the waste sulphide liquor. I shall explain to you the process. There is the steaming process through which we can remove the sulfur dioxide, then we can condense back this sulfur dioxide and we can have liquid liquor that is totally free from sulfur dioxide. The propagation conditions are similar to Baker's yeast fermentation process. pH of the media is about 4 to 5 depending on the raw materials. Temperature is 25 to 30 degrees centigrade. Aeration is essential. This is also aerobic fermentation process. Foaming must be controlled with the help of anti-foam oil. So, these are the fermentation conditions we required for fodder yeast fermentation process. This is kind of that flow chart that we have, how the waste sulphide liquor can be used for the fodder yeast production. So, here we can have the blended tank here. Then I shall explain this stripper. The stripper, we pass the steam so that we heat the material. When we heat the material, sulfur dioxide goes out, and sulfur dioxide and water make the sulfurous acid. Then you cool this liquid, and it doesn't contain much of sulfur dioxide, and then we add some kind of nutrient to it and take it in the fermenters, and after that it produces that the cell mass. Then we pass it through the primary separator, then washing stage, then refrigerate the cream tank, and drum dry for drying purpose, and the flakers, and finally cyclone mixer, we get the dry yeast cells. Now this is the drum dryer. I already explained how drum dryer functions. It inclined at a certain angle and it rotates at very low RPM. I can explain this like this. Suppose you can put the material here, and I told you it will be like this. Your material should not be free falling. It should be some kind of addition there. So this is rotates. This rotates like this, and this is here we have some kind of gear arrangement. We have here we have motor with the help of this is grounded with this, and here some kind of supporting materials. We have this also kind of grounding. So it is rotated. So here we pass the dry air. When here you have wet material or wet biomass, when it goes like this, your material comes in contact with the dry air, and dry air will be going like this. Here, this is you have wet air. You will get some kind of wet layer outside. So it goes in process. When your material comes here, it will be dry solid. This is how it functions. It's largely used by the industries. I explained this process during when I explained the citric acid fermentation process. Now, fodder yeast, the raw materials is filtered through the 80 mesh stainless steel skin mounted on a wooden blend tank, 125,000 gallon capacity. I already discussed what you mean by 80 mesh. 80 means that means the port size is approximately 1 by 80 inch. This is the port size of the mesh, that that sieving mesh is about that 1 by 80 approximately, not exactly. The hot liquid is pumped from the building tank to the top of the steam stripper, which reduces the sulfur dioxide content. I explained that how it is done. The stripper is 5 feet diameter and 20 feet tall. So it is very simple that we can have. This is like this. We can pass here steam we can pass, and here you have the liquid, that is sulfide. So here we will get the sulfur dioxide, and here we get the liquid. This is like this. So stripper, this is the diameter is 5 feet, and this is 5 feet, and how tall is 22 feet. So this is like this. This is how it is used, and steam is that admitted the stripper through the sparger. So you have sparger here through the drills. We can have the sparger, they equally distributed through this, and sulfur dioxide and water vapors are condensed in the condenser and returned to the paper mill for use. It is again recycled back to the paper mill for reusing purpose. The recycling is there. So here it can be explained. Here it is waste sulfur liquor is coming here, steam is coming here. This is a stripper. Then vapor of sulfur dioxide and water, it condenses here. So you get the sulfurous acid, because both are the coolers. here also you cooling this, and here also hot liquid also cooling, and this liquor is good for the fermentation process. We reduce the temperature to 25 to 26 degrees centigrade. Now, the strips sulfite liquor passes through the bottom of the still through the control valve automatic level type at 80 degrees centigrade. It is cooled to about 26.7 degrees centigrade by passage through the stainless steel coolers admitted to the fermenter, and then the liquor contains 1.5% sugar, but it is it has the poor nitrogen, phosphorus and magnesium content. The phosphorus is added in the form of calcium dihydrogen phosphate. This is something you can correct it. Ca is to PO. See, this is okay. Ca is to PO4, and this is this is calcium dihydrogen phosphate, or this is potassium dihydrogen phosphate, ammonium dihydrogen phosphate. Nitrogen is admitted in the form of ammonium sulfate. And I told you, you can remember this ammonium sulfate purposefully added in case of Baker's fermentation process to keep the pH of the media acidic, because here also in case of Fodder fermentation process we required the pH 4 to 5. Now, Fodder production, the fermenter constructed in carbon steel is clad with stainless steel open at top and 26 feet diameter and 14 feet height, equipped with the central drop tube 4 feet in diameter. Air is supplied in the bottom of the drop tube through the modified valve of aeration wheel, which possesses open end tubes. Now, wheel rotate at 300 rpm. This air is supplied by the centrifugal blower at the rate of that 1700 cubic feet CFM. CFM is the cubic feet per minute, and that the wheel is in combination with the drop tube acts as a mechanical defoaming agent. The oat containing the yeast cell suspension moves downward in the interior of the drop tube and upward around the exterior. So, this is the typical feature of the Fodder yeast fermentation process that we have. The nutrient that is disodium hydrogen phosphate, potassium iodides are mixed with the water in 500 gallons tank and pumped to the head, that is the surge tank, because surge tank is located in the higher level, and they are fed continuously by gravity to the fermenter from the head tank. The aqueous solution of ammonia containing about 20% ammonia is also fed continuously into the fermenter at a rate which is correlated with the sugar content of the sulfite liquor. The pH of the oat is about 5, and fermentation is carried out at 37 degrees centigrade. A cell and tube heat exchanger is used to control the temperature. This is, we required some kind of heat exchanger to control the temperature of the liquid. The oat is removed from the one side of the bottom of the fermenter and passed through the heat exchanger and returned to the other side of the fermenter. The process is continuous one in which the contamination has not been a problem. Since this is a continuous process, some of the yeast suspension is removed continuously, and yeast is then filtered in 3 stages to separate the solid and wash separately, wash repeatedly, and this is then dried in the drum dryer to obtain the desired moisture content, and then it is sold. The yeast, the yield of fermentation product is about 50 to 53% on the basis of sugar fermented. So, this is more or less this is all about this yeast fermentation process, and what I try to mean here that the Torula utilis is used as a fodder yeast, and it has the typical characteristics because it can degrade not only the hexose sugar, but also it can degrade the pentose sugar. And this contains some kind of nucleic acid. That is why it is recommended that as a fodder. And fodder means the animal system, they can easily utilize this nucleic acid containing of that biomass very easily. So this can be used as a good fodder. And since it is a animal feed, that is we use some kind of waste material for the production of this fodder yeast, and we find that sulfide waste liquor that is produced from the pulp and paper industry, that is found to be the best raw materials for this fermentation process, because pulp and paper industry that usually we use the bamboos as a raw materials, or some kind of lignocellulosic material is a raw materials for the production of paper, because paper is nothing but it is pure cellulosic material, and this undergoes some kind of digestion process. So the lignin removal that take place. When we remove the lignin, then it takes out some kind of hemicellulose present in the bamboos, that also comes out. And this hemicellulose, I already pointed out, it comprises of 80% of hexose sugar and 20% of pentose sugar. So that is why we find it is a very good material. But when we try to use this material, we find that they use in the pulp and paper industry, we use the sulfur dioxide as the oxidizing agent, because this bleached the paper. Because when we see our paper, it should be perfectly white, and the bleaching is done with the help of sulfur dioxide. So sulfur dioxide content in the sulfide waste liquor is quite significant amount. So this is to be removed before we use for the this fodder yeast fermentation process, and this can be removed by steam trapping technique. In the steam trapping technique, what we do, we pass the stream and to heat the liquid to about 80 degrees centigrade or more than that, so that vapor of sulfur dioxide and water goes out from the top, and the liquid which is coming out from the bottom, it doesn't contain much of sulfur dioxide, and it can be we supplement this with some nitrogen and phosphorus source. We already pointed out that for the growth of microorganisms, we required nitrogen source, we required carbon source, we require minerals and vitamin. So then when those materials you have to put desired amount, so that your growth of the organism that can take place to the desired extent. And this is another thing I told you, this is like Baker's yeast fermentation process, this is also aerobic fermentation process. So you have to do kind of aeration for this process. The one speciality of this fodder yeast is that when you dry this fodder yeast, it gives a meaty flavor. That is the typical feature, typical characteristics we have as per fodder yeast concerned. So we covered both the Baker's yeast fermentation. Baker's yeast is a food yeast, and fodder yeast is a kind of animal food for the can be used. So this is greatly used in day-to-day life. Thank you very much.